Hello everyone, today we have the first lab called pGLO transformation, and it is the first lab in the series of GFP transformation and expression labs. In this experiment we are going to do the bacterial transformation, in which bacteria Escherichia coli K12 will uptake the circular piece of foreign DNA or plasmid. We'll do the heat shock transformation, which means that we are going to put our bacteria at 42 degrees for 50 seconds. The plasmid we are going to use is pGLO, which contains the gene coding for GFP or green fluorescent protein. It allows the glowing of bacteria under the UV light. The bacteria we are going to use are non-pathogenic, but K12 strain requires the use of standard microbiological practices, which means that we will need to decontaminate surfaces with 70% ethanol before the work. For this experiment we would need a starter plate with Escherichia coli grown, and you can see the single colonies on it. You would also need port agar plates, agar, which makes a solid support for bacteria to grow, 2 plates with both LB agar and ampicillin, the antibiotic that is going to be a selective factor for transformed bacteria, and finally the last plate with LB agar, ampicillin and L-arabinose sugar, which will initiate the expression of the GFP. We would also need the transformation solution, which is 50 millimolar calcium chloride with a pH of 6.1, the pGLO plasmid, and LB broth, the liquidous medium for growing of bacteria. We will also need the ice bucket, pipettes, a waste beaker, pipette tips, 70% ethanol for surface decontamination, and also the inoculation loops, which are disposable plastic inoculation loops here. Bacterial transformation will take place in the water bath, which is set for 42 Celsius degrees for this experiment. Label 2 Eppendorf tubes, one with plus pGLO sign, another with the minus pGLO sign. Add 250 microliters of transformation solution to both of the tubes. Always when you start the experiment, make sure that you mix the solutions well. Since the tubes are sterile, we don't need to use 2 different tips for the tubes. After you dispose the tips, place the tubes on the ice bucket. Using a disposable inoculation loop, pick up a single colony from a starter plate. Do this gently because you do not need to dip it into agar. Just lightly touch it and then immerse the loop into the tube labeled plus pGLO. Make sure that you mix it well so that the bacteria are separated. Repeat the procedure for the minus pGLO tube. Touch the colony gently and transfer it to the minus pGLO tube. Mix it and dispose the loop. Pipette 10 microliters of pGLO plasmid into the tube with the plus pGLO sign. Mix by pipetting up and down several times and do not add DNA into the tube with the minus pGLO sign. When you pipette it up and down, do it gently because the forced pipetting up and down will make vortex, which might harm your DNA content. Incubate the tubes on ice for 10 minutes. Now it's time to label your plates. Do it on lid, not on the bottom. So for the plus pGLO sign, label LB ampicillin and LB ampicillin arabinose plates. For the minus pGLO sign, label LB ampicillin plate and just LB plates. For the heat shock, you need to transfer the tubes to the water bath set to 42 degrees for 50 seconds precisely. After 50 seconds, transfer the tubes to the ice bucket and place it for 2 minutes. Add 250 microliters of OLB broth to both of the tubes. Incubate the tubes for 10 minutes at room temperature. Pipette it up and down before you transfer the LB broth to the tube. Also, do not put much force to pipette down the LB broth, because it might create bubbles, and bubbles will trap bacteria and DNA. Do the same for the second tube. Now add 100 microliters from plus pGLO tube to the plates labeled with plus pGLO sign. Before you take 100 microliters, pipette it up and down gently. Make sure that everything from the tip gets into the agar plate. Trash the pipette tip and use an inoculation loop, evenly distribute the suspension of the cells on the surface of agar. Even distributions allows the place for the cells to grow. Repeat the procedure using another inoculation loop. Now transfer the minus pGLO tube solution into the plates with minus pGLO sign. You're going to pipette it up and down. Take 100 microliters, transfer them to the plate, remove the tip, and repeat for the next plate. Next, evenly distribute the suspension of the cells on the surface of agar using the inoculation loop. Make sure you inactivate your waste after you finish the experiment. Now transfer all of the plates to the incubator and place them upside down. This is going to be 37 degrees incubator. Place it overnight. Clean your bench and the experiment is done.